ವಿದ್ಯಾರ್ಥಿಗಳೇ, ಕಳೆದ ಕೆಲವು ಸಂಚಿಕೆಗಳಲ್ಲಿ ನಾನು ಪ್ರಿನ್ಸಿಪಲ್ಸ್ ಆಫ್ ಇನ್ಹೆರಿಟೆನ್ಸ್ ಬಗ್ಗೆ ಮಾತಾಡಿದ್ದೆ. ಕೆಲವು ಟಾಪಿಕ್ಸ್, ಇನ್ನೂ ಕೆಲವು ಟಾಪಿಕ್ಸ್ ಇದ್ದಾವೆ, ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸಲ್ಲಿ ಮಾಡ್ಕೊಡ್ತೀನಿ ಅದನ್ನು. ಈಗ ಈ ಕ್ಲಾಸಲ್ಲಿ ಮಾಲಿಕ್ಯುಲರ್ ಬೇಸ್ ಆಫ್ ಇನ್ಹೆರಿಟೆನ್ಸ್. ಇನ್ ಬೋತ್ ದ ಚಾಪ್ಟರ್ಸ್ ಯು ಕಮ್ ಅಕ್ರಾಸ್ ವಿತ್ ಟರ್ಮ್ ಇನ್ಹೆರಿಟೆನ್ಸ್. ಇನ್ಹೆರಿಟೆನ್ಸ್ means it is the transferring of the characters from the parents to their progeny. Inheritance. Okay. Heredity is also transferring of the characters from one generation into another generation. Heredity. Then what is the difference between those two, heredity and inheritance? Inheritance explains the transferring of the characters from parents to the offspring in a particular manner. How the characters are transferred from the parents to their offsprings. Okay. So inheritance explains the way of, pattern of transmission of the characters, genetical characters from the parents to their offsprings. Right. So in that chapter we studied the genetics at the, in that chapter we studied that chapter genetics principles. Right. In this chapter we study about the genetics or inheritance of characters at the molecular level. Okay. At the molecular level. You see, in this chapter you are going to study about the nucleic acids. What are nucleic acids, how they are functioning, etc. So in this chapter there are so many topics are there. Let us discuss one by one. Okay. Let us discuss about the nucleic acids. Nucleic acids. Friedrich Miescher was the greatest scientist. He discovered the nucleic acids in a pus cell, right? But he didn't know that these materials are the nucleic acids. So he called that materials present inside the nucleus was the nuclein. The materials which were present in the nucleus were the nuclein. Later on, the later scientists renamed that nuclein as nucleic acids. This nuclein is called nucleic acids because of two reasons. Number one, these materials are present in the nucleus. Number two, these materials exhibit the acidic properties. Because of these two reasons, this nuclein was renamed as nucleic acids. Okay. So as I told you, these nucleic acids were discovered by that Friedrich Miescher. Right. In this nucleic acid there are two types. One is the DNA and another one is the RNA. I think you know the full form of this DNA and RNA. RNA, ribose nucleic acid. DNA, deoxyribose nucleic acids. So these are the two nucleic acids present in the nucleus primarily. Okay. Whatever may be the nucleic acid, whether that nucleic acid is a DNA or RNA, basically that nucleic acids is formed of three components. One is the sugar and the second one is the nitrogenous bases and third one is the phosphate group. So these are the three components present in any nucleic acid, whether you consider the RNA or DNA. For example, components of RNA. This RNA consisting of the sugar present in RNA is called as the ribose sugar. Ribose sugar. Remember that this ribose sugar is a pentose sugar. It means it consisting of 5 carbon atoms. So the formula of the ribose sugar is a C5H10O5. So this is a sugar present in RNA. Like that, the same sugar present in DNA also. But here it is called as the deoxyribose sugar because in that sugar present in DNA having C5H10O4. In this same sugar, one O atom is deleted. That's why this sugar is called deoxy, removal of oxygen, deoxyribose sugar. So Sugar present in RNA is called as the ribose sugar. Sugar present in DNA is called as the deoxyribose sugar. Of course, both of them are pentose sugar with 5 carbon atoms each. Right. Concerned with the nitrogenous bases. In each case, there are 4 nitrogenous bases. For example, the nitrogenous bases present in RNA, nitrogenous bases present in RNA are number 1, adenine. It is designated as A. Number 2, guanine, G. Number 3, cytosine, C. Last one is the uracil, U. These are the 4 nitrogenous bases present in RNA. And these 4 nitrogenous bases again can be classified into 2 groups. Both this adenine and guanine together called as the purines. Purines. Like that, this cytosine and this uracil together called as the pyrimidine. Pyrimidine. So that in the nucleic acids there are 2 groups. One is the purines and another one is the pyridines. Okay, pyrimidines. Both adenine and guanine are called as the purines and the cytosine and uracil are called as the pyrimidines. Usually purines are larger and dicyclic. Pyrimidines are smaller and monocyclic. Right? Okay, so these are the 4 bases, nitrogenous bases present in RNA. Like that, in DNA also there are 4 nitrogenous bases. They are number 1, adenine, guanine and cytosine. These 3 nitrogenous bases are common both in DNA and RNA. But the last nitrogenous base is differed. In case of RNA, it is uracil, while in case of DNA, it is the thymine. It is a thymine or thiamine. So that the nitrogenous bases present in DNA are adenine, guanine, cytosine and thiamine. These are the nitrogenous bases, right? So phosphate group, it is common in both the cases. Okay. These are the components, basic components present in the nucleic acids, right? And these nucleic acids actually forming the nucleosides and nucleotides. Okay. Then what are nucleosides? Nucleoside is a combination of one pentose sugar with any one nitrogenous base. So this is nucleoside. So if the question is asked what is a nucleoside, you have to answer like this. Nucleoside is a combination of a pentose sugar with any one nitrogenous base. Nucleoside. As there are 4 bases in RNA, in DNA, we can see the presence of 4 nucleosides. In case of RNA, there are 4 nucleosides. In case of DNA, there are 4 nucleosides. They are, for example, nucleosides of RNA. Nucleosides of RNA. Number 1, adenosine. Number 2, guanosine. Number 3, cytidine. Number 4, uridine or uretidine. Okay, uridine or uretidine. These are the 4 nucleosides present in RNA. For example, how this adenosine is formed? This adenosine is formed by the combination of ribose sugar plus adenine. Ribose sugar plus adenine gives rise to adenosine. So adenosine is a nucleoside of RNA. Like that, this guanosine, ribose sugar plus guanine. Ribose sugar plus guanine gives rise to the guanosine. So guanosine is a nucleoside. Like that, this cytidine is formed by the combination of ribose sugar plus cytosine. Like that, this uridine is formed by the combination of the ribose sugar plus uracil. These are the 4 nucleosides present in RNA. Okay. So, if the question is asked for 2 marks, what is a nucleoside? Give an example. You have to write the definition and one example. Sometimes the questions will be asked, mention the nucleosides of RNA. Then you have to answer like this: adenosine, guanosine, cytidine and uridine. These are the 4 nucleosides present in RNA. Okay. Like that, in case of DNA also, there are 4 nucleosides. There are 4 nucleosides. They are, for example, number 1, deoxyadenosine. Deoxyadenosine. Number 1. Number 2, deoxyguanosine. Deoxyguanosine. Number 3, deoxycytidine. Number 4, deoxythymidine. These are the 4 nucleosides present in DNA. And these nucleosides are, for example, this deoxyadenosine is formed by the combination of deoxyribose plus adenine. This one is formed by deoxyribose plus guanine. Okay, deoxyribose plus cytosine, deoxyribose plus thymidine. So these are the 4 nucleosides present in DNA, right? So this is about the nucleosides, both in RNA and DNA. And these nucleosides with the phosphate forming the nucleotides. Nucleotides. Then what are nucleotides? Nucleotides are the combinations of nucleosides plus phosphate group. Okay. Or you can say nucleoside is a combination of one pentose sugar, one nitrogenous base and one phosphate group. Nitrogenous base. Okay. So the combination of these 3 together called as the nucleotide. Right. So in case of RNA, there are 4 nucleotides. For example, AMP, adenosine monophosphate. Adenosine monophosphate. Okay. Single phosphate. Adenosine monophosphate is a nucleotide. If this nucleotide consisting of 2 phosphates, ADP, adenosine diphosphate. If there are 3, adenosine triphosphate. Like that. Okay. AMP is a nucleotide. GMP is a nucleotide. CMP is a nucleotide. UMP is a nucleotide. Right? I hope you have understood this like this. So, AMP, adenosine monophosphate. This adenosine monophosphate is formed by the combination of, by the union of adenosine plus phosphate. GMP, guanosine monophosphate. It is formed by the combination of guanine plus phosphate, cytidine plus phosphate, uridine plus phosphate. Okay. If there is only one phosphate at least with this nucleoside, then that will become nucleotide. So, nucleotide is a combination of nucleoside plus phosphate, or you can say nucleotide is a combination of one pentose sugar, one nitrogenous base and minimum one phosphate. Okay, nucleotide. So, in case of DNA also, there are 4 nucleotides like in case of RNA. Okay, DAMP, DGMP, DCMP, where it is a DTMP. Deoxyribose thymidine, deoxyribose thymidine monophosphate. Okay. So, like this, there are 4 nucleotides in RNA, in DNA, like that. Okay. So, you see, nucleotides are formed. Nucleotides are formed, as you know, by the combination of one sugar, one phosphate, okay, and one nitrogenous base. The combination of these 3 is called as one nucleotide. You see, for example, adenosine monophosphate is a nucleotide. Adenosine monophosphate is a nucleotide. This adenosine monophosphate is formed by the combination of one pentose sugar. This is the pentose sugar. For example, this is ribose sugar. I told you this ribose sugar is a 5-carbon structure. The first carbon is this. This is the first carbon. This is second carbon, third carbon, fourth carbon. This is the fifth carbon. Okay. This pentose sugar, when combines with a base, nucleoside is formed. You see, that base is always attached to the first carbon, actually to the OH group of this carbon. For example, adenine. Adenine, okay, is a base. It is attached to the first carbon of the sugar. Now this combination is called as the adenosine. Right? Adenosine. And this adenine mean this base is always attached to the first carbon of the pentose sugar by means of the bond. This bond is called as the glycosidic bond. The bond present between the base and the pentose sugar is called as the glycosidic bond. Right? And remember that any base is always attached to the first carbon of the pentose sugar only. First carbon of the, right? That too to the OH group of the first carbon. This is one nucleoside. And you see, to this nucleoside, if one phosphate, minimum one phosphate is attached, it will become nucleotide. Remember that phosphate is always attached to the fifth carbon of the pentose sugar. For example, this is. Now this is the nucleotide. Now the bond present between the phosphate and the fifth carbon is the ester bond, or you can say phosphoester bond. Phosphoester bond. Okay. So these are the two bonds formed in the nucleotide. So phosphate is always attached to the fifth carbon by means of the bond. It is called as the phosphoester bond. The base is always attached to the first carbon of the sugar by means of the bond. That bond is called as the glycosidic bond. This is one nucleotide. Okay. Then you see, when we consider any RNA, any DNA, any nucleic acid is formed by means of polynucleotide chain. This one nucleotide was I wrote there, one nucleotide. Like that, in the RNA chain, RNA strand, there are so many nucleotides. So RNA strand is a polynucleotide chain. Like that, the DNA also polynucleotide chains. So both RNA and DNA are formed of nucleotides, so that they are the polynucleotide chains. Okay. So this is a brief information about the components of the nucleic acid. Okay. Now let us study about the structure of the DNA. Structure of DNA. You see, in 99.9% cases, DNA is the genetic material. DNA is the hereditary material. It is called the genetic material. It is called hereditary material because it carries the genetical characters from generation to generation. So it is called the hereditary material. But RNA is also genetic material, but only in some cases. RNA becomes genetic material where there is no DNA. RNA kuda genetic material ಆದರೆ ಎಲ್ಲ ಕಡೆ ಅಲ್ಲ. DNA ಎಲ್ಲಿರುತ್ತೆ ಅಲ್ಲಿ DNA is the genetic material, no doubt. ತಿಳ್ಕೊಳ್ರಿ, DNA ಇದೆ ಅಂದ್ರೆ ಆ ಸೆಲ್ ಅಲ್ಲಿ ಅಥವಾ ಆರ್ಗಾನಿಸಮ್ ಅಲ್ಲಿ ದಟ್ DNA is the genetic material. ಬಟ್ RNA ವಿಚಾರಕ್ಕೆ ಬಂದಾಗ RNA is also a genetic material. ಆದರೆ ಎಲ್ಲಿ ಅಂದ್ರೆ where there is no DNA. Where there is no DNA, where the RNA becomes the genetic material. For example, there are so many viruses are there. RNA virus. In RNA virus, no DNA. So that RNA becomes the genetic material. It is like that. ಹಾಗಾಗಿ ನಾವು ವೈರಸ್ ಅಲ್ಲಿ RNA ವೈರಸ್ DNA ವೈರಸ್ ಅಂತ ಕ್ಲಾಸಿಫೈ ಮಾಡ್ತೀವಿ. DNA ವೈರಸ್ ಅಂದ್ರೆ ಇನ್ ದಟ್ ಕೇಸ್ DNA is the genetic material. ಅಲ್ಲ RNA ಇರುತ್ತೆ, ಬಟ್ ಅಲ್ಲಿ RNA ಅದು ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್ ಅಲ್ಲ. RNA ವೈರಸ್ ಅಲ್ಲಿ RNA ವೈರಸ್ಸಲ್ಲಿ RNA ಮಾತ್ರ ಇರೋದು. ಸೊ RNA ಇಸ್ ದ ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್ ಲೈಕ್ ದಟ್. ಓಕೆ, ಸರಿ. ಈಗ ಈ DNA ಇದೆಯಲ್ಲ, ಹೇಗಿದೆ DNA? ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ DNA ಹೇಗಿದೆ? ಯು ಸಿ, ನಂಬರ್ 1, ದ ಫಸ್ಟ್ ಪ್ರಾಪರ್ಟಿ ಆಫ್ ದಿ ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ದಿ DNA. ಈಚ್ DNA ಮಾಲಿಕ್ಯೂಲ್ ಮತ್ತೊಂದು ವಿಚಾರ ನೆನ್ಪಿಟ್ಕೊಳ್ರಿ. ಈ DNA ಎಲ್ಲಿದೆ ಅನ್ನೋ ವಿಚಾರನ ನೀವು ಫಸ್ಟ್ PUC ಓದಿರ್ತೀರಿ. ಎಲ್ಲಿದೆ DNA? ಯು ಸಿ, ಲಾಸ್ಟ್ ಇಯರ್ you have studied the cell structure, different organelles of the cell. While studying the different organelles, you have studied the nucleus. Nucleus actually containing the chromosomes. Contain the chromosome. For example, if this is the cell, if this is the nucleus, inside this nucleus we can see so many thread-like materials. They are the chromosomes. Then what are chromosomes? Chromosomes are the longer, thinner, microscopic, thread-like colored bodies. Carry the genetical characters from generation to generation. ಹಾಗಂತ ಕ್ರೋಮೋಸೋಮ್ಸ್ ಬಗ್ಗೆ ಡೆಫಿನೇಷನ್ ಓದ್ತೀವಿ. ಕ್ರೋಮೋಸೋಮ್ಸ್ ಅಂದ್ರೆ ಏನು ಅಂದ್ರೆ, ಕ್ರೋಮೋಸೋಮ್ಸ್ ಆರ್ ದಿ ಲಾಂಗರ್, ಥಿನ್ನರ್, thread-like, microscopic thread-like colored bodies present in the nucleus. They carry the hereditary characters from generation to generation ಅಂತ ಓದ್ತೀವಿ ಕ್ರೋಮೋಸೋಮ್ಸ್ ಬಗ್ಗೆ ಓದ್ಬೇಕಾದ್ರೆ. ವಾಟ್ ಆರ್ ದಿ ಕಂಟೆಂಟ್ಸ್ ಆಫ್ ದಿ ಕ್ರೋಮೋಸೋಮ್ಸ್ ಅಂದ್ರೆ, ಇನ್ಸೈಡ್ ಈಚ್ ಕ್ರೋಮೋಸೋಮ್ DNA is the main material present. DNA ಎಲ್ಲಿದೆ ಅಂದ್ರೆ, DNA present inside the chromosome. ಹಾಗಾದ್ರೆ ಕ್ರೋಮೋಸೋಮ್ ಅಲ್ಲಿ ಬರೀ DNA ಮಾತ್ರ ಇದೆಯಾ? ನೋ, ಕ್ರೋಮೋಸೋಮ್ ಅಲ್ಲಿ DNA is the main material. In addition to that, in the chromosome we can see the presence of RNA to some extent, some proteins, etc. ಹಾಗಾಗಿ DNA ಲೊಕೇಶನ್ ಎಲ್ಲಿ ಅಂದ್ರೆ, DNA is present inside the chromosome and that chromosomes are present inside the nucleus, right? Come to the structure of the DNA. Each DNA molecule is formed of 2 polynucleotide chains. Each DNA molecule is formed of 2 polynucleotide chains. There are 2 chains. There are 2 strands. There are 2 DNA strands in each molecule. Okay? And that chain, that strand is a polynucleotide chain. So, that chain is made of many nucleotides. So, in each molecule, there are 2 such chains are there. So, the first point what you have to write is about the property. Each DNA molecule is formed of 2 polynucleotide chains. Second point, and these 2 polynucleotides are arranged in opposite manner and they are twisted with each other and they are forming the spring-like structure. Okay? For example, this is the structure of the rough structure of the DNA. This is structure of DNA. In this DNA, there are 2 strands, you see, and each strand is a polynucleotide chain. So, in each molecule, there are 2 polynucleotide chains. Okay? And both strands are twisted with each other to form a double helical structure. Double helical structure. This is the second point you have to write. Okay? First point, this is the first point. Each DNA molecule is made of 2 polynucleotide chains. Okay? Each chain is helically coiled. ಪ್ರತಿ ಥ್ರೆಡ್ ಇದೆಯಲ್ಲ, ಅದು ಸ್ಪ್ರಿಂಗ್ ಇದ್ದಾಗಿದೆ. ಅದು ಅಷ್ಟೇ ಅಲ್ಲ, 2 ಥ್ರೆಡ್ ಒಂದಕ್ಕೊಂದು ನೂಲ್ಕೊಂಡಿದ್ದಾವೆ. ಸೊ ದಟ್ ಈಚ್ ಸ್ಟ್ರಾಂಡ್ ಈಸ್ ಹೆಲಿಕಲಿ ಕಾಯಿಲ್ಡ್. ಅಟ್ ದ ಸೇಮ್ ಟೈಮ್ both the strands are coiled about each other to form a double helical structure. This is a second point what you have to write while explaining the structure of the DNA molecule. Right. Third point, you see, ದ ಬ್ಯಾಕ್ ಬೋನ್ ಆಫ್ ಈಚ್ ಸ್ಟ್ರಾಂಡ್. ನೋಡಿ, ಇದು ಒಂದು ಥ್ರೆಡ್ ಅಂತ ಅನ್ಕೊಂಡ್ರೆ, ಒಂದು DNA ಸ್ಟ್ರಾಂಡ್ ಅಂತ ಅನ್ಕೊಂಡ್ರೆ, ಇದರ ಒಳಗಡೆ ನಾವು ಒಳಗಡೆ ಭಾಗವನ್ನ ಕೋರ್ ಅಂತ ಕರೆಯೋದು ಅಥವಾ ಬ್ಯಾಕ್ ಬೋನ್ ಅಂತ ಕರೆಯೋದು. ದ ಬ್ಯಾಕ್ ಬೋನ್ ಆಫ್ ಈಚ್ ಸ್ಟ್ರಾಂಡ್ ಕನ್ಸಿಸ್ಟಿಂಗ್ ಆಫ್ ರಿಪೀಟೆಡ್ ಶುಗರ್ ಫಾಸ್ಫೇಟ್ ಎಟ್ಸೆಟ್ರಾ. ಅಂದ್ರೆ ಇದರ ಒಳಭಾಗ ಇದೆಯಲ್ಲ, ಥ್ರೆಡ್ನ ಒಳಭಾಗದಲ್ಲಿ ಏನಿದೆ ಅಂದ್ರೆ ಶುಗರ್ ಇದೆ, ಅದೇ ಪೆಂಟೋ ಶುಗರ್. ಅದ್ರ ಪಕ್ಕದಲ್ಲಿ ಫಾಸ್ಫೇಟ್ ಇದೆ, ಅದ್ರ ಪಕ್ಕದಲ್ಲಿ ಶುಗರ್ ಇದೆ, ಪಕ್ಕದಲ್ಲಿ ಫಾಸ್ಫೇಟ್ ಇದೆ, ಶುಗರ್ ಇದೆ, ಹಾಗೆ ಉದ್ದಕ್ಕೂ ಹಾಗೆ ಇರೋದು. ಅದೇ ಥರ್ಡ್ ಪಾಯಿಂಟ್. ಸೊ ದ ಬ್ಯಾಕ್ ಬೋನ್ ಆಫ್ ಈಚ್ ಸ್ಟ್ರಾಂಡ್ ಈಸ್ ಫಾರ್ಮ್ಡ್ ಆಫ್ ರಿಪೀಟೆಡ್ ಶುಗರ್ ಫಾಸ್ಫೇಟ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್. ಯು ಹ್ಯಾವ್ ಟು ರೈಟ್ ದಟ್ ಪಾಯಿಂಟ್. ಓಕೆ. ಸೊ ಒಂದು ಇಲ್ಲ ಬರೀ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಇದಾವೆ. ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಅಲ್ಲಿ ಶುಗರ್ ಸಿಕ್ತು, ಫಾಸ್ಫೇಟ್ ಸಿಕ್ತು, ಮತ್ತೆ ವೆರ್ ಇಸ್ ದ ನೈಟ್ರೋಜನಸ್ ಬೇಸ್? ನೆಕ್ಸ್ಟ್ ಪಾಯಿಂಟ್ ಅದು. ಸೊ ಥರ್ಡ್ ಪಾಯಿಂಟ್ ಏನ್ ಬರೀಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ, ದ ಬ್ಯಾಕ್ ಬೋನ್ ಆಫ್ ಈಚ್ ಸ್ಟ್ರಾಂಡ್ is formed of repeated sugar and phosphate molecules. Sugar and phosphate, okay? This is sugar and phosphate molecules. Fourth point, the nitrogenous bases are always present inner to the structure of the molecule. ಇದು DNA ಮಾಲಿಕ್ಯೂಲ್ ಆದ್ರೆ ಇದು ಒಳಭಾಗ, ಅದು ಇಲ್ಲಿ ನೈಟ್ರೋಜನಸ್ ಬೇಸ್ ಇರ್ತವೆ, ಇಲ್ಲಿ ನೈಟ್ರೋಜನ್ ಬೇಸ್. ಸೊ ದ ನೆಕ್ಸ್ಟ್ ಪಾಯಿಂಟ್ ಇಸ್ the nitrogenous bases are always present inner to the structure of the molecule. For example, this is sugar, phosphate. This is sugar. You know that the nitrogenous base is always attached to the first carbon of the sugar. So that nitrogenous base present here, nitrogenous base like that. So this is the fourth point. So the nitrogenous bases are always present inner to the structure of the molecule. This is the next point you have to write. Then the next point, this nitrogenous bases forming the base pairs. This nitrogenous bases forming the base pairs. That nitrogenous bases forming the complementary base pairs. Then what are base pairs? What are complementary base pairs? You see, for example, just I will write this. Consider that these two are the DNA strands. The same strands are written here, but in straight line, okay? But they are always present in this manner only. DNA molecule ಯಾವಾಗಲೂ ಇದೆ, ಹೀಗೆ ಇರೋದು. ನಾನು ನಿಮಗೆ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ಲಿಕ್ಕೋಸ್ಕರ ಇದನ್ನ ಸ್ಟ್ರೈಟ್ ಬರೆದಿನಿ ಅಷ್ಟೇ. ಈ ತರ ಸ್ಟ್ರೈಟ್ ಇರೋಲ್ಲ ಅವು, ರಿಮೆಂಬರ್ ದಟ್. ಓಕೆ, ಇದು ಶುಗರ್ ಅಂತ ಅನ್ಕೊಳ್ರಿ, ಇದು ಶುಗರ್. ಅದು ಇಲ್ಲಿ ಬೇಸ್ ಇರ್ತವೆ, This is phosphate, this is sugar, phosphate, sugar, okay, sugar, okay. Consider that ಇಲ್ಲಿ ಮಧ್ಯೆ ಫಾಸ್ಫೇಟ್ ಇದೆ ಅಂತ, ಇಲ್ಲಿ ನೈಟ್ರೋಜನಸ್ ಬೇಸ್ ಇರ್ತವೆ, ಎಲ್ಲಿ ಶುಗರ್ ಇರ್ತವೆ ಅಲ್ಲಿ ನೈಟ್ರೋಜನಸ್ ಬೇಸ್ ಇರ್ತವೆ, ಆಯ್ತಾ? ನೋಡ್ರಿ, these nitrogenous bases forming the base pairings or base pairs, don't worry, okay? Base pairings, they are forming the complementary base pairings. ಹಾಗಂದ್ರೆ ಏನು? ಬೇಸ್ ಪೇರಿಂಗ್ ಅಂದ್ರೆ ಏನು? ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಬೇಸ್ ಪೇರಿಂಗ್ ಅಂದ್ರೆ ಏನು? ಪೇರಿಂಗ್ ಅಂದ್ರೆ, ವಾಟ್ ಈಸ್ ಬೇಸ್ ಪೇರಿಂಗ್ ಅಂದ್ರೆ, it is a pairing of the nitrogenous bases. Okay, it is a pairing of the nitrogenous bases in the DNA molecule where the purine is always paired with the pyrimidine. So base pairing andre, it is a pairing of the nitrogenous bases where the purine of one strand is always paired with the pyrimidine of the opposite strand. This is base pairing. For example, consider that this is adenine. This adenine is a purine. Okay, this is a pyrimidine. So this is base pairing. So base pairing is it's a pairing of the nitrogenous bases in which the purine of one strand is always paired with the pyrimidine of the opposite strand. This is base pairing. Then what is the complementary base pairing? Complementary base pairing is also a base pairing, but it's a specific base pairing in which the A is always paired with T only. Like that, this G is always paired with the C only. This is C, this is G. this is a T, this is A, like that. This is complementary base pairing. So complementary base pairing is one in which the adenine is always paired with thymine, and this guanine is always paired with cytosine. So that complementary base pairing andre, it's a specific base pairing in which the A is always paired with T and G is always paired with C, and vice versa. This is complementary base pairing. Okay, ಆ ಪಾಯಿಂಟ್ ಬರಿಬೇಕು. ನೆಕ್ಸ್ಟ್ ಏನು ಬರಿಬೇಕು? ಅಂಡ್ ದಿಸ್ between these nitrogenous bases, the pairing occurs because of the presence of the hydrogen bonds. Okay, mention the bond present between the nitrogenous bases ಅಂತ ಕ್ವಶನ್ ಇದ್ರೆ ನೀವು ಹೈಡ್ರೋಜನ್ ಬಾಂಡ್ ಅಂತ ಬರಿಬೇಕು. ರೈಟ್, there is always a double hydrogen bond between AT and triple hydrogen bond between GC. That you have to know. Okay, double hydrogen bond between AT and triple hydrogen bond between GC. Actually, they are the weak bonds. But in the textbooks, weak bonds are not shown. Directly it is shown as the continuous bond. So this is the actual bonds present between the nitrogenous bases. Weak hydrogen bonds. But no problem. If you write hydrogen bonds, it is enough. Okay. So the nitrogenous bases present always inner to the structure of the molecule. They are forming the base pairings and they are forming the complementary base pairings. Right. Next point. You see, from here to here, it is called one turn. This is another turn. Another turn. Each turn usually consisting of 10 base pairs. Each turn generally consisting of 10 base pairs. For example, this is one pairing, another pairing, another pairing, another pairing, another pairing, like that. From here to here, we can see the 10 base pairings in a normal DNA molecule. There may be DNA which are having 9 base pairings, 11 base pairs, like that. But according to our textbook or what we are studying in our textbook, having the 10 base pairs in each turning. Okay, remember that. And the distance between two base pairings is about 0.3 nanometer. So that the length of the one turn is 3.4 nanometer, or you can say in angstroms it is 34 angstroms, like that. So these are the important points concerned with the structure of the DNA molecule. Right. So the DNA is always present in the double helical structure, while RNA is always present in a single helical structure, single thread like structure. This is a difference between those two, mainly regarding the chain structure. Right.